Hello again, everyone. Welcome to Q&A with A&V. I'm Vincent Racaniello, and joining me today from Columbia University in New York City, Amy Rosenfeld. She's on my left there. Hi, Amy.
Hi, Vincent. How are you today?
I'm well. You have a lovely
Good.
blue sky out the window there.
Uh, Maybe it's the polarization. It's kind of looking like it's gonna open up any minute and let
Oh,
that
it's going to
shower.
rain? Really?
I think so. Smells like rain.
Before we get
You know?
started, yeah, I know that smell, but do you get it in the lab? Yeah, they do pull the outside air in, right?
Yeah, are we discussing the big news?
What big news? The lease? Yeah, but first I want
Yeah.
to show a picture here that I forgot to show last week of Amy doing plaque assays. If you thought she wasn't serious, this is what she does every day, right, Amy?
Yes, I do. The incubators can no longer put stuff into them. They're gonna vomit stuff out. Yeah, that's me.
Also, this week on Monday, oops, that's the wrong one. On Monday, Daniel and I signed a lease on something 7th Avenue. I forgot the address. It's 2 blocks south of Penn Station. We signed a 5-year lease on studio office space. I have a little video. Do you think it'd be okay to show it, Amy?
Yeah, I think it's
It's pretty
exciting.
short. Okay, watch this, folks. So I just did this on my camera just to give you an idea of the space. Um, so they're going to now do renovations. They're going to knock those walls down, build that wall up, take the rug out, put a new rug in, paint the walls white, knock the other walls down. It's going to take about a month, I guess. And then I will be going there to work every day and making more content for you folks. All right, that's pretty exciting. And I have to say, this young lady sitting next to me is really the one who moved this forward because 2 years ago I said, Daniel and I want to rent a studio. And of course, we didn't do anything. So one day she got in touch with a realtor and moved it forward. That's how Amy works. All right, let's check out what's going on here in the world. There have been some early chatter. I'm going to go just to Vincent. Any questions with Vincent or Amy in front? Because I know the rest are you guys chatting amongst yourselves right now. There are 200 plus of you, which is cool. And if you don't have Vincent, here we go. Let's start with Red. Nicholas Wade was on the Quillette podcast. He seems unaware of any quality criticism. Yeah, I mean, they don't ask me. They ask Nicholas Wade. Fine.
Who are they?
I have no idea.
Okay, well, then I don't think we can be insulted because I don't know who they
He
is.
seems unaware of any quality criticism. What world is he living in? Unbelievable. Unbelievable. Now, I'm getting a lot of
I'm
pushback.
sure he's aware of I'm sure he gets pushback. I'm
Well,
sure he just blows it off. It doesn't bother him.
all right, I'm going to make a video where I go through every point and rebut it because everything he says is rebuttable, okay? It's going to take me some time, but that's what I'm
Well,
doing
now
now.
you have plenty of time.
Well, I don't have a studio yet, but I can still do it. Now, earlier we were talking about MERS coronavirus, which of course comes from, it occurs mainly in the Arabian Peninsula, goes from camels to people. And I said if we vaccinated the camels, we could stop it. You don't have to vaccinate people because not enough people get it, maybe a few a month.
But it
And
doesn't really go people to people.
yeah, it doesn't go
It's
people to
like
people. That's right.
every time there's a people person who gets infected, it's a new spillover event.
That's right. So this person says, Deborah, too bad we couldn't vaccinate the bats. Right. That would be very difficult. Camels are kept captive, right? So you can easily
Yeah, they're
vaccinate
the
them.
transportation or the pets or something or other.
Susan really enjoyed the Gary video. So we did on Twiv in 1 week 2 videos, one with the WHO committee, 3 people, one with Gary. Many of you like them, but also many people tell me it was a big disappointment for them. For this reason and that reason, blah, blah, blah. I don't care what you think about it. They're the good videos. That's the truth there. Oh,
Well,
boy.
they should just listen to Twiv on Friday when you talk about Eddie Holmes's paper that I sent you today.
Yes.
That just really like now you have to sit back and you have to say, really? You're gonna go with this lab leak hypothesis, really? Are you on drugs? If so, send them my way.
And Mike says, is this study total bunk? SARS-CoV-2 RNA reverse transcribed and integrated into the human genome. Yes,
Total
it's
shit.
total bunk. It's wrong. It's an artifact. Other viral RNAs.
The guy never should have been allowed to publish it.
No, it's being shown by others now that it's an artifact. No
It's
SARS-CoV-2 RNA has ever been found as DNA in the genome of any patients.
It's 4 papers now that have wasted money because of this man's hubris.
Skip questions about origin. They irritate Amy. Is that a good plan, Amy?
Sure. Well, I don't think that that's a good plan.
How are your plaques doing today?
We got plaques, so it was kind of a disappointment because we want no plaques.
Okay, the Pfizer dose plan for under 12 is about half that of 12 and up. How much does vaccine dose size make for a 12-year-old? I don't understand the question. Do you, Amy?
No.
I don't know what science went into the thinking of making it half the dose. But, under
Maybe
12.
because they're less. Maybe because they're less mass.
Yeah, size is smaller, so,
Yeah. So.
Drugs are given per kilogram sometimes, so you could imagine the same.
Mm-hmm.
And as I said, folks, put my name or Amy's in front, so that I will answer just those because I know you're having a chat. Ivermectin, RCT trials don't target early replication, while quasi show 50 to 70% reduction. Okay, so on the TWIV that's dropping tonight, 766, we had David Fagenbaum on who talked about this, and he said, the RCTs that have been done are too small to make any conclusion, but they say it's probably worth doing a large RCT of 4,000 people, and he said that's going to be done. All right. Now, the key is, of course, it has to be done early because if it's an antiviral, you have to give it early in infection. If you wait till people are hospitalized, it's going to be too late, and it's not going to show efficacy. Here's another one. Maybe Amy knows. How much animal surveillance has China been doing since the pandemic started? We have no idea. But the paper that we will do Friday on TWIV is all about bat surveillance in China, right? And also in other countries as well.
Yeah, but they're Australian, right?
Yeah, they're not from China. So we don't really know what's going on. All we hear are vague things like, surveillance continues. But, they do publish. There's no question about that.
I mean, the truth of the matter is we've pissed them off so badly. Why do they want to help us?
Yes. And we continue to piss them off by our position. I shouldn't say our, but the position of some that the virus came from there.
Yeah, I mean, you don't walk into a lab or an office and say, "This is how you're going to do it." Like, there's no respect.
I have to say, this week I sent an email to Zhongli Shi, who is a coronavirologist at the Wuhan Institute of Virology, and
She's the bat lady.
The bat lady, and I said, Zhongli, I just want you to know that there are some virologists here and in the world who know that this virus came from nature and not a lab, and I hope that gives you some comfort. And she wrote back and said, thank you so much. I appreciate what you do on TWIV. And she said, I really wish people would look at the science and the data and realize we have a bigger problem to address here, which is getting rid of this pandemic. But she seemed to me just like a person like we are, She's not hiding anything.
No, but I do like the fact that she wrote about how she listened to TWIV.
Yes.
And I think it's admirable that under all of the scrutiny that is unjust that has been given to her, the tone of her response was her still belief in humanity and science. You can imagine that you would be disgruntled and somber and depressed, but that wasn't the tone of the wording, right?
Yep. Nope. And I wanted to make her feel better because can you imagine much of the world thinking the virus came from her lab? Can you imagine how you feel? And it's not true. So I tried to give her some comfort, and she seemed to like that. And it was just a normal person virologist like us. This is for Charlotte. I'm sorry. Aussie Cam. S proteins from African isolates were nearly fully virulent. Other studies show that West African camel isolates carry mutations in MERS-CoV, which may limit human transmission. So this is about MERS coronavirus. Okay. This is talking about isolates from camels. I'm not aware of that. I'm sorry. Is Mexico's low case count due to seasonality? I don't know. It could be as we move into summer. It could be due to vaccination. It could be due to immunity from infection.
Could be due to not testing.
Not testing. Yeah. If you already recovered, should you take a vax if it's offered? Yes. Absolutely. Here we go. V&A. Victoria and Albert. There's a famous hall in London called the V&A. Did you know that, Amy?
I did, but I've never been to London, so I've never been.
But it's the opposite of A&V. Due to temporal reaction, they're thinking myocarditis might be a biological component. Any insight to help me decide best choice for my 12-year-old twins?
Daniel. Listen to Daniel last Friday. He told you he had a 15-year-old. His son was in the, heart of that group. He said that myocarditis is transient, very rare. He vaccinated. Listen to Daniel.
Okay, V&A. Say I'm fully vaccinated but still get infected and develop symptoms, however mild. Does this mean I'm more likely to pass it on than if I remained completely asymptomatic?
No.
Not at all. No relationship. At all. Is there a large ongoing clinical trial? As I said, it will be done. There's David Feigenbaum said a 4,000 person RCT will be done. I did not go to lunch. It was canceled because of a scheduling conflict. We're going to get through the emails quickly because there's no things in the front. Why do people say COVID-19 is seasonal when a lot of hot countries like India are having problems? Well, let me tell you, Genster 1, we say influenza is seasonal in temperate climates that have a winter. And a lot of countries, they have a winter season, but it doesn't get very winterish like it does here in New York. So it can be seasonal in some areas, but maybe not in others. But flu is also seasonal in the tropics, by the way, and we don't understand why. So I suspect, that SARS-CoV-2 could be seasonal in hot countries, and if so, and we don't know yet, there's a reason that we're not aware of. Blue Mountains, Ontario. Hey, hi from Lebanon. Oh, this is great. Love it. Love to hear it. What is Amy listening to? Do you listen? Oh, in the hood in your picture. Here. What are you listening to there in those earbuds, Amy?
Probably older episode of TWIV. Or the last episode of TWIV. It depends.
Sometimes you listen to music, though, right?
Sometimes I listen to music. Sometimes I listen to different podcasts. Sometimes they're just don't talk to me. I'm not in a chatty mood.
Okay, Marty. Vincent, wondering if you saw the study done by the Cleveland Clinic saying those with past COVID do not need to be vaxxed. I do not agree with that, and that is because Daniel Griffin has said you should be vaxxed because we are not aware of the extent of natural immunity and its longevity after natural infection. Now, this study is a small one, and I don't think it's definitive. And I don't know what the point is. We have enough vax to go around. And do we see today that the U.S. is going to provide some 100 millions of doses in the next few years? Is that good news, Amy?
I think it's great. I think it's great that we're buying like 100 million doses of Pfizer to give away.
Amy, Mr. Ozzy wants to know why you think SARS-CoV-2 and MERS couldn't recombine.
Well, they're both beta coronaviruses. They're at the opposite. They're at the opposite extremes
Of the
They're in
phylogenetic
different
tree, yeah.
Yeah, they're in different groups, so they cannot combine.
Very unlikely, you mean, right?
Yeah.
Okay, with the number of replication cycles over the past 18 months, how likely is it that the ancestral will be found? By ancestral, do you mean in bats, for example? Yes, we can still find it because, remember, the mutation rate so the problem is that right now we have many, many millions of people infected, but in bats there's not millions of people infected with the ancestral virus, so the mutation rate is much slower. So yes, we should be able to find it. And there'll be markers, genetic markers that you can use to show that it is the ancestral strain, but mostly it'll be percent genome identity, right?
Yeah, I mean, they should tune in for Friday's TWIV. Paper is very clear.
All right, so the water cooler's going. We're gonna put what do you call that soda stream thingy in the kitchen, right?
Yes.
Because Amy said she won't visit unless there's a soda stream.
There must be bubbles. I like bubbles.
Yes, we will tear down the wall. The building owner is tearing down the wall.
The building owner is at the head of his own construction company.
Yeah. How soundproof is the office? Well, the neighbors are offices. There's no noise. The only noise, there's the air conditioning, which I can deal with, and I will put some soundproofing up. And, of course, the windows. When a fire truck goes by, it'll be noisy. So I may buy sound blankets to put on the windows when I'm recording, unless I want the light, in which case we'll have to deal with the noise when it comes by. But I think it'll be great. I'm very excited to set it up, and from scratch. I'm going to get all new stuff, so your donations will be very much appreciated. And that way I can do more content. I can make multiple videos a week in addition to podcasts. And I'm hoping to do, as I said, live stream virology courses in the future from there. So I have a lot of plans, and to hire other people if we can raise money. So Microbe TV is becoming a nonprofit. If we can raise money, I can hire people to help out. So I have big plans. But as Amy says, unless I move forward, I'm not going to go anywhere, right?
Right. But donations aren't going to be enough. You need to have a little soiree with the big people.
Yeah, we have to work hard to... Should I hire someone to be a fundraiser, Amy?
Sure.
Okay, Rock of Ages, Nature paper delaying Pfizer second shot. 3.5 higher antibody levels, but lower T cell. Age 68, don't leave my home. B1672 worries me as I live in the California fire area. I just don't think that delaying is going to affect T cell response. It doesn't make any sense to me.
I think he's fine. He doesn't have to go to a hotel if he doesn't want to, but I think he can go visit family or whatever outside of the California fire area.
Yeah. By the way, it's Amy's incubator. Yeah, so it'll be called the incubator. So I want to get signs for the door. If anyone knows where I can get signs made, I only want one of each, not 40 or 100. Every place online I look. So we want one that says the incubator. We want one that says Parasites Without Borders, and one that says Microbe TV, to stick on the door with our logos. If you know a good place to order them, send it to me. Amy's Incubator. That's good. Should we call it Amy's Incubator? No. What are your thoughts on gain of function?
We do gain of function every day.
Yes.
We're for gain of function. Worth potential risks? Do you get in your car? Is your driving your car to work worth the potential risk of being hit and killed?
Yeah, so as Amy said, everybody doing microbiology research does gain of function. And if you stopped all gain of function, you would really have no research, very little research. If you take something that you put in food, say, a compound that you put in food, and it's a genetically engineered compound, and you want to modify it to make it more stable or more active in enzyme, that's gain of function. Some gain of function experiments that involve viruses could result in viruses with different pathogenicity or tropism, and for those you have to have the right containment, and they're regulated. So gain of function with SARS coronaviruses, avian influenza viruses, as an example, they're regulated, and you have to do them under high containment, and that's fine, and they are worth the risk because they tell us, for example, what restricts the transmission of avian influenza viruses, or for coronaviruses, what could make them infect human cells. Thank you, Les, for your super chat. Go Incubator. Thanks, Amy.
You're welcome. I don't know. I just think that all this high and mighty questioning of gain of function, everybody gets in their car. Everybody takes not everybody, but most people take some kind of birth control or some kind of medicine, or they voluntarily have voluntary surgery. Every time you unnecessarily cut or have some medical procedure, you have a potential chance of dying. Is it worth it? Is the potential chance of dying worth it for your facelift?
As someone said later in the chat, gain of function does not mean creating a killer virus. It's a general name for many kinds of experiments, but the press has given it a bad name.
It's not the press. It's not. Unfortunately, it wasn't.
Who is
it?
It
was Albright and the idiot at Harvard.
Ebright and Mark Lipsitch.
Yeah, it's them.
How does the SARS-CoV-2 RNA get its cap, Amy?
It has a capping enzyme.
Yeah, the
It
virus
has a
encodes a
capping
capping enzyme.
enzyme. It has a capping complex. So there's a methyltransferase that takes it from SAM and complexes it, and then with the capping enzyme, it caps the mRNA.
Thank you, T. Klupper, for your contribution. You know, everyone, we really appreciate your coming here and your contributions. Really make us recognize that you appreciate what we do. HZH, could you please comment on the protection effectiveness of natural immunity versus vaccine? Oh my gosh, a reporter sent me this question today because he found a link on Facebook that said one is always better than the other. Now I don't even remember what they were saying.
That's not true.
But it's not. It depends on the virus.
Totally. Yeah. I mean, for HPV, the vaccine is better, but for other cases, it's not. And actually, I think that the paper that I sent you from Viviana and Kramer say that the vaccine in some cases is not better than natural immunity for SARS-CoV-2.
The fact is that we don't know at this point for SARS-CoV-2. And if anyone tells you they know, they're not going by the data.
A year, it's their paper. They looked at their cohort a year later.
Yeah, but that's pretty soon, right, Amy?
Yeah, I'm just saying that I wouldn't be so harsh. They have a cohort that they've been following and stuff, and from their cohort, this is their conclusion.
Wow, you're telling me not to be harsh.
Well, I mean, it's an
overinterpretation.
It's an overinterpretation.
What caused the sea change when talking about lab leak?
Who's sea change?
All of a sudden it's back, right? The Wall Street Journal did it a couple of weeks ago with the dumb article which had no truth in it and no science.
Yeah, but it didn't come from the Wall Street Journal. It came because the WHO meeting was discussing, the meeting of the committee was presenting its findings to the general meeting that week. That's why it came back.
Yeah, that's right. That's what Marion Koopman said, right, when we asked her.
Yeah, it had nothing to do with the schmagegi at the Wall Street Journal. He just was an opportunist.
Rock of Ages, how do you donate? Well, you can go to microbe.tv/contribute, and you could do PayPal or Patreon. And if none of that works, you could send me an email, and I'll give you an address to send a check, our new office address. And then as they come in the morning, they'll be under the door. Because I said, where do you put the mail? And he said, they just leave them under the door. I have to get a little mailbox.
Okay.
Glad to catch you live with my girlfriend who's a first grade teacher and enjoys your content as much as I do. Oh, that's lovely. Thank you so much. It's great. Is anyone searching for bat guano for SARS-CoV-2? Sure, absolutely. But the problem with bat guano is, right, it's all mixed together from many different bats. And a better way is to get individual bats and see what they have, right? To get a sense of what's in the population. Right, Amy? Ooh, I did the T-shirt thing.
Yeah.
Did you get your T-shirt, Amy?
No.
Oh, that's the wrong one. Every time I say, right, Amy, I should bring the T-shirt up. Well, next, when you get yours, are you going to wear it one night on the live stream?
Maybe. I don't know. I haven't decided. I think actually that, what's his face? Eddie Holmes. I think that they do look at bat guano in that paper. And I think the problem isn't that it's from all the bats. I mean, it doesn't really make a difference. It's from all the bats in that cave. It's the problem. It really doesn't. It's really the problem that there's so very little virus.
Yeah, right. Hey, Amy, someone later in the chat says, all week, Carl, he said, all week I've been wondering if Vinny remembered the CO2 needed changing.
I had to remind him, but tomorrow he's bringing bleach, and I have to remind him too.
So I pick up bleach.
Morning.
So we use bleach in the lab to inactivate viruses. It's very good for that, right? If we order it through a lab supplier, it's $50 a gallon. And so I can get it at Acme for $5. So I pick it up. Isn't that ridiculous? $50 a gallon for bleach? So
well, it's like $100 for a thingy of aluminum foil.
Yeah, you know, the big roll of aluminum foil, which you would have at a deli, right? You pull it off and you rip it
Yeah,
off.
it's like $100.
I could probably get that from Amazon. Maybe I should be ordering everything, right?
Yeah, but I don't need aluminum foil now.
Why are viruses host specific? Why didn't they develop a genetic makeup that can work in a wide range of hosts and target common receptors? They don't have to. They all have individual hosts and niches. And why they're host specific can be receptors, but also beyond the receptor in the interior of the cell, all the proteins that are needed can be host specific. But a virus doesn't have to be pan specific because it can find a host in a little nematode in the soil in a vineyard in France, and that's good enough for it. Anything to add, Amy?
No?
How bad will the India variant, that is the Gamma.
I thought you just told me it was the Delta.
Sorry, Delta. My apologies. I don't like the nomenclature. I think it sucks.
Well, who can remember it?
Well, you have to remember the order in which they occurred. Alpha is UK.
Yeah, well, that doesn't have any.
Beta is South Africa. Delta is... Gamma is Brazil, and Delta is Indian. Anyway, how bad will it get? It won't get bad because this variant, as far as I can see, is handled by vaccination. And it makes up a very small proportion of cases in the U.S. right now. It may displace other viruses, but that's because it would be slightly more fit, but it's going to be handled by the vaccine. So people just get vaccinated and you don't have to worry about the variants.
So, did you see the paper that I put in the folder by somebody that we know, but I forget who it is.
Raul?
No, it's not Raul, but it's somebody. Oh, it's by Nevin. It's by Nevin and Adolfo and I forget who is the actual senior author on the
What
paper.
does it say?
So they look at one of the variants, whatever. Let's just call it variant A. And it turns out that all the hoopla of the spike mutations don't do dick. It has to do with the fact that it has an alteration in ORF9A, which is an interferon antagonist. Oh, and it's super active. All this hoopla. So it's more fit. It replicates better because it is better at turning off interferon.
It's not in the folder. Can you check later?
I'm sure it's in the folder.
All right, tomorrow.
All right, fine. I'll look at it.
Or there are a ton of good papers. There's one on multi-inflammatory syndrome. There's the nanobody one, which is really
the nanobody's like the Peter Walter one. I wonder where Peter's thing is.
There's also another one I passed. Oh, so many good papers, so little time. Okay, back to the chat here. Okay.
Yeah, I just thought it was funny because it got written up by Carl Zimmer and in the Times, and he doesn't even have the courtesy to come out and basically say, I was a jackass. I was totally wrong when I wrote all these articles about how it was more transmissible and everything. I should have waited for the science, but I didn't.
Amy's been saying for a year that the other changes in these variants, not just the spike, make a difference, and now it's turning out to be true.
Yeah. Well, I said for a year that the spike variants don't do anything because it doesn't make any sense to be tighter to the receptor.
Yeah.
You have to come off at some point. It doesn't make any sense, right? So, yeah. And why you couldn't just start off your article with, I was wrong.
No, people don't do that. Scientists don't like to say they were wrong.
He's not a scientist. He's a journalist.
So journalists, in fact, especially, never say they're wrong.
Well, he was wrong.
Yeah, they never admit it. Absolutely never. If you know an example, tell me. By the way, folks, last week you complained that my image was bad. It's better now. Right? I fixed my LUT. I went back and fitutzled with the camera. Is that the word, Amy? Fitutzled?
Yeah, fitutzled.
All right. 2 weeks after my second Moderna, I plan to ride the Toronto subway a lot, hoping to get modest inoculation to the Delta variant. Is this a dumb thing to do? Yeah, you don't need to. You're fine. You're protected against disease with that vaccine. You don't need to But in the course of moving through your life, you may get infected and you may get boosted. You wouldn't even know it. That's how vaccines work. Vincent, India is not a randomized clinical trial, but it sends a signal. I would argue it says that maybe we should do an RCT, and so it's being done. So we'll see if ivermectin works in an RCT. But, you know, if the results are negative, people will still say, but it works in India. So I don't know what to do about that.
Well, it's not clear what it's working against, right?
Are Australian studies a problem? Did you say they were? Or maybe it's another Amy, because I don't remember.
Why are Australian studies a problem?
I don't know.
Please
Please
explain
explain if you're an issue. If you explained it on Twitter, please reference. All right, so I would say the Robert Gary episode, right? We did the Furin site pretty well.
Yeah, but I think that actually you guys have all overlooked one little tidbit of information.
What's that?
I believe that avian influenza, when it can acquire a furin cleavage site normally.
Yeah, it does. That's when it becomes highly pathogenic. Yeah.
Right. So, and that's all in nature. So to argue that a virus can't acquire a furin cleavage site or a protease cleavage site is totally wrong.
TWIF 762 Bad Cat with Robert Gary. He explains it really well. Is there anything new in the Delta variant we haven't seen before? No, it is. It has two spike changes that have been seen in other variants, and it's infecting a lot of people because it's in India, which has a huge population, and so people normally think, it must be a bad variant because it's infecting so many people. No, they have 1 billion people, and they're mostly not vaccinated. You put any SARS-CoV-2 in there, it's going to be a problem, right? And this is what nobody's thinking about.
Well, again, you only spoke about the spike variants. I guarantee you that there's other alterations within that genome.
Actually, there's an ORF8 change.
Okay. Perfect.
Thank you, SL, for your super chat. Really appreciate it, your support. Your support means a lot, especially because a lot of people say we're useless. Yes, I will. So I will make that video. I'm going to make a gain of function video, and I make sure to run them past Amy first, and she'll tell me if it's good or not, ready for prime time, right? Well, thank you, Vern, for your wonderful contribution. Whatever you guys can contribute. I hear thunder. Oh boy, and I'm in the basement. You got thunder there, Amy?
No, but I got a new umbrella.
But anyway, any of you come to New York, you're welcome to visit and see what we've put together.
Yes, but don't come to the lab. We don't take visitors.
Yeah, I saw this paper. I don't believe it in the least. First of all, it's a preprint, and I think it's going to be changed after peer review. So there are modest changes in responses to innate triggers. You can give...
Shocking.
What? Shocking, yes. And it's not at all clear what they mean. They didn't do a control, which was try another vaccine and see if it does the same thing. So I don't think...
Or why don't they just do an empty nanoparticle? Yeah. The lipid thing. I guarantee you half of those changes are due to the lipids.
Yeah.
Because the lipids are sensed.
I think it's not a good study. It doesn't mean a lot, yet the press picked it up. It's a preprint. The press picked it up. They didn't know what to do with it. And they asked a few people, what do you think of this study? And they didn't read it. And they go, oh, wow, that could be important, without reading it. And then, wow, that could be important, gets propagated through the webs, and that's how this stuff happens. You know, part of the problem is they ask people for their opinion on a paper, and then the people give an opinion by just reading the abstract. And you can't do it. You have to read the whole paper.
Oh, really?
And if people out there think I'm being too harsh, well, that's science, folks. And journalism and science are sometimes like oil and water. You know, there's some very nice writers out there, but I don't think they get it all the time. Greetings from Buenos Aires. Christian Drosten did a podcast about the virus origin. He comes down on the side of natural origin. Very good. I'm glad to hear it. It came from nature. And you'll see in 10 years or whatever it takes, if I'm still around, you'll see it came from nature. Vincent, you have a good heart. Touched by your email to the Chinese virologist. Thank you very much. I thought she needed some support. And Amy will tell you, sometimes I can have a good heart, right?
Yeah, for sure.
It's right, Vincent, not simply right. I guess it could be right, Vincent, but nobody says right, Vincent, right?
No.
Hello, Esther. Thank you for your work. My question is, I'm of the understanding the virus can remain latent within the body. So if someone, and it continues, who is vaccinated contracts the virus and that virus remains dormant within the body, if the vaccine loses its effectiveness, what do you think the chances are that the coronavirus revives itself and begins to attack the body? I want to take that, Amy?
It doesn't lay latent in the body. It doesn't reactivate. It's not herpes.
Right. So, Esther, some viruses remain latent, but not all. And herpes viruses are the big ones, and there's some others. But SARS coronaviruses, influenza viruses, polio viruses, enterovirus D68, none of them remain latent. They come and go.
They're acute viral infections.
Esther, you ought to listen to my virology videos on YouTube. They're free. And you can go right to the one that is about acute infections, and then the other one is persistent infections that contrasts the two different kinds of infection patterns. Or you don't have to. I know they're an hour each. Just take it from us. Someone wrote an email today, Amy said, thank you for TWIV. I tell so many people to listen, and I don't understand why they don't. Because it's hard. People don't like stuff that's hard. You guys here, of course, all 459 of you. Wow. Can you imagine? 459 people have come to listen to two virology nerds. It's amazing. Faster Toiters 2, thank you for your contribution. Not all heroes wear capes or do plaque assays. It's great. I love it. Is there any way to make a condensed version of TWIV? Yeah, it's long, I know. Maybe that's...
Well, that's one of the ideas, is that you're going to have a 15-minute thingamabobber once a week that just does the highlights.
So I could take the bigger one, the longer ones, and edit it to be really tight, right?
Yeah, or yeah.
Or I was thinking of making a separate one, but maybe I'll edit it. I'll try editing it. What can we call it?
Twivettes.
Twivettes or Twivlets or something like that. Yeah, good. That's great. Good idea. Is there some talk about pan-corona vaccines? Are there preclinical indicators that can be used to rule out the possibility of ADE for a pan-corona vaccine? What do you think about that, Amy?
I think you did Barrick's paper on the potential of generating a pan-corona vaccine, and there was another paper at the same time. Listen to TWIV. So
you could do animal studies to look at ADE. First, you look in cell cultures with Amy. Amy doesn't like those experiments, but
it's total crap.
With ADE, you see if a virus could get, into a cell with an FC receptor via an antibody bridging the FC receptor in the virus. And if you get that, then you could do a mouse model for it. But Amy's not a big fan of those.
No. All right. Well, 2 more questions because it's already 8:45.
2 more questions, and then this is what happens?
Yes.
Not so worried about myocarditis as much as it coming from LHC-MISC, multi-inflammatory syndrome. Yes. Well, I agree. You ought to listen to...
POTS.
Positional orthotopic, right? POTS.
I don't know. What is it?
POTS.
I don't know. It's got
postural orthostatic tachycardia syndrome, where a change from lying to standing causes an abnormally large increase in heartbeat. POTS. That's a lot of ME/CFS patients have POTS. You ought to listen to TWIV 766 coming out today with David Feigenbaum. Man, he talks about Castleman's disease, which is a cytokine storm disease. Scary as hell. And it relapses. You get it every couple of years and no clear treatment.
No, but Carlos says that some of this isn't cytokine storm stuff. It's other stuff.
One more question for Amy. Let's find a good one. And then we will come back to... I lived in the tropics. I never heard of anyone. Well, there is flu in the tropics, Deborah. You may not have heard of it, but really, because you didn't hear of it, you think it doesn't happen? I don't mean to be insulting, but there are tropics in the flu. I can show you the papers. Here we go. Do viral vector vaccines exhibit SARS-2 epitopes on the modified ad or only SARS genetics?
What?
Explain how the adenovirus vectored vaccines work.
So the spike protein is encoded within the genomic material that's packaged in the adenovirus. And then when the adenovirus infects the cell, because you have a cell line that makes the particle and you have the packaging signals all on the DNA, when it infects the target cell, it uncoats and releases the genomic material. The DNA is transcribed into mRNA. The mRNA is then translated. And the protein is then processed and glycosylated just like it would be in the context of natural infection.
Yeah, so it's
I don't
genetic.
know why. I don't understand the question.
Well, she wanted to know if the epitopes were on the adenovirus. So no, they're not. It's the,
no.
as Amy said, the spike gene is incorporated into the adenovirus. Thank you, Amy.
Thank you. Okay, bleach. And I forgot now, but bleach and some others. Yeah, bleach and some, I forgot now, some other stuff tomorrow.
Okay. Bye, Amy.
Bye.
Okay, let me turn Amy off there. All right. And let's get back to our chat here. Simon, thank you for your donation. Oh, thank you. Taking the time, letting the WIV lab know they are just normal, good, hard-working scientists. Thank you. I'm glad some of you appreciate it. Here's about transmission and humidity. The viral season is more related to humidity than to heat. It's a combination of the two. In the Northeast, when it's cold and low humidity, you get better transmission of influenza virus. But in the tropics, it is not cold, and the humidity is not as low as it is in, say, the northeast of the U.S. So it's not just that. It may be that the transmission is different. It may be more contact. We don't know. Yes, I know the humidity does vary, but that's not it. That's not the answer. Tom, thank you for your support. Really appreciate it. I'm glad you folks like this. Most of you like it anyway. Some of you do not. I'm sorry about that. I'm learning to deal with it. Is there reason to believe some virologists might be biased against lab leak because it's negative for the profession? You mean like me? No. I'm not worried about the profession. The profession will survive. Let me just say one thing. I'm driven by data. The lab leak hypothesis is not. That's all you need to know, folks. I don't want to hear any other conspiracy stuff. It's about data. There are no data to support it. End of story. End of story. I'm suffering with a really bad sore throat. It's not COVID. Makes me wonder if traditional infections are hitting us harder after all the distancing. I think hard to know, Carl. It's hard to know what's going on. It is if you're in the northern hemisphere, you're in the summer months now, right? So we do still have respiratory virus is continuing to circulate. So you could have that happening. Are there any currently known mutations not covered? The mRNA vaccines, as far as I know, protect against disease caused by the main variants of concern: alpha, beta, delta, gamma, and that's all I need to know. And people say, "What do I do about the variant?" Just get vaccinated. If you are terrific, get your second shot. If you're not, go get vaccinated. Please do. Thank you, Powell, for your contribution. Really appreciate it. In the Philippines, some establishments require face shields. So face shields are typically needed in together with a face mask in, say, a medical establishment where you have a lot of patient contact. Although I was in one today, and there was no such face shields. So I think for the man on the street or the woman on the street, you don't need a face shield. You just need a face mask. So I think that requirement is not really, doesn't make any sense to me. I'm a programmer, but my chimp brain predicts Novavax and mRNA vaccine combo will provide excellent immunity. What do you mean, together? I don't think you need to put them together. mRNA on their own are fantastic. And it could be that Novavax will be very good on its own as well. And we don't know yet because we don't see the phase 3 results. So, and they're both nice because you could reprogram them very quickly, right? You could make new mRNA vaccines. You can make new protein-based vaccines. Am I going to use a green screen? I don't know. I used the green screen when I recorded the parasitology lectures. Over on my YouTube channel, there's a whole series of lectures on parasitology by Dixon and Daniel, right? And I used a green screen for them, and I put, images behind them, but I don't think they used them very well. I think if you've ever seen those iBio series seminars, they have green screens where then they put slides in the background and the people point to them, but I always think it's artificial. If I'm going to use a slide, I will put the slide on and just show that. You don't need to see me while I'm showing the slides. I mean, it's worth trying. I'm always open to new things, right? Jan, how transparent is the military? Military's gotten more transparent. They've actually started to support a lot of basic research, because as you know, they have a good deal of money, and they have started to fund research that is relevant to the health of service people. And of course, much there's an overlap between that and us, the public. So you could develop vaccines and so forth. So a good deal of it is, I'm sure a good deal of it is classified, and I don't know about it. So remember, if you want to ask me a question, put my name in front, because I know you guys have conversations, and I want to skip through those. I got AstraZeneca first and just got Moderna. We'll have super immunity. Not super. You'll have really good immunity. I don't think there's super immunity. Coffee mugs with Amy and Shocking. Shocking would be a good one. Absolutely. I think that would be great. Okay, we have... What's your biggest concern about the threat from SARS-CoV-2? My biggest concern, believe it or not, is not having enough people vaccinated, because as soon as 70 to 80% of the world is vaccinated, circulation will drop, variants will drop, and this will no longer be an issue. Vaccination solves all the problems, folks. Nothing is going to emerge that will be a problem if we're all vaccinated. And all it's now is a matter of making enough vaccine for everyone. There's no magic virus that's going to arise and poke through the vaccine. It's not happening. The press would like you to think that. I see zero evidence for that. Now, the monoclonals, if you have to be treated with monoclonals, then the variants have a problem, because some of the monoclonals don't work against the variant. So you need to know, if you have a patient, say, who gets sick, gets COVID and needs to get a monoclonal therapy, you need to know what variants are circulating in the area because you have to give them the right cocktail of monoclonals. That's a situation where it's important. But that's a fraction of the world's infections. Vaccines are really going to take care of the rest. Has this crisis accelerated science's understanding of viruses? No, I don't think that we have accelerated a fundamental understanding of viruses, okay? In fact, I think we've forgotten a lot of the fundamentals because a lot of people have come into COVID and are not virologists, and they're saying things, well, how long will this immunity last? Well, why don't you just look at the literature and you can see for other viruses and other vaccines how long it lasts. Most of the answers are out there. And so I don't think we have learned an awful lot because of that. I think we've regressed to a large extent. Excuse me. I'm a little disappointed in the way. Now, on the one hand, the field has responded big time. It's great. So many people have tried to contribute to solving this problem. But on the other hand, all this nonsense about variants, transmission, virulence, the origins, this is a result of people not looking at history. So I'm disappointed. And if you say what's the most significant new understanding, we knew all the fundamentals before. There's nothing new learned here. We made vaccines really quickly, and that's great. We made monoclonals really quickly, but we don't have any quick antivirals, do we? So we still have that to learn. And I don't mean to be angry at all. I want to tell you my disappointment in science in a way that so many bad papers have been published that are just downright wrong. For example, taking organoids from the brain and saying, oh, they're infected with SARS-CoV-2, therefore SARS-CoV-2 must infect the brain. This is nonsense. What happens in a culture does not predict what happens in a real brain. You have to do the experiment. And there's no evidence that the virus is reproducing in the brain. And by the way, we're going to have a neurologist on in a few weeks to talk about that. Daniel Griffin tweeted that some PCR tests may no longer be effective, mutations in spike. Well, of course, if the change in the RNA target is altered, remember, in the original UK variant, there was a deletion that caused the PCR not to work, and they eventually figured it out and dealt with it. So I'm not too worried about that. The Indian variant is claimed to be more transmissible. Let me show you a quote from Ron Fouchier, okay? Let's share my screen here. Here we go. So this is from my show notes from TWIV 765. I do a Google Doc, so here you go. You're seeing the sausage being made. I have show notes. And I do not, this is a quote from Ron Fouchier. I do not think that any of the SARS-CoV-2 variants is truly more transmissible nor more infectious or contagious. These are all words being used, which are probably all wrong, or at least there is no evidence for such phenotypes. For the D614G variant and later the B117, aka British variant, there was evidence for efficient global spread, outcompeting prior variants, but that is not the same thing. Look at that. That is not the same thing. This is not me speaking. This is another virologist who knows about transmission. We see this for flu all the time. Such displacements can even be the result of neutral genetic drift. For flu, as you know, this is often due to immune escape. Antigenic variants can infect a slightly larger number of hosts, and that is enough natural selection advantage for the new variants to outcompete the old ones. I attach a slide deck where I presented this, and I don't have it here, to show that flu variants replace each other like SARS-CoV-2 without anyone ever talking about increased transmission. Host population is changing, not the virus, in my opinion. And then I wrote, trying to get Ron on TWIV, and he's agreed to come on to talk about this. So in every case that a new variant emerges, there is no proof of better transmission. The variants are more fit. That means they reproduce better. And what Ron said is a virus only needs to be slightly more fit. What makes it more fit? Many things. One of them is evading antibodies. You evade antibodies a little, which we know these variants do. They spread better than an other virus in a population, and they simply outcompete it. What happens in India, we have an outbreak for other reasons. And the variant's there, and it's infecting. It doesn't mean the outbreak has any special relation to the variant. It's just in the right place at the right time. So that's what I mean by more fit. There can be many mechanisms to be more fit. I think of it another way. Do you really think that more transmissible variants are arising in every country all over the world now every month? That's crazy. It's never been seen before in virology. This virus was more transmissible when it first emerged. These are more fit viruses. The evidence presented for better transmission is very circumstantial. It's not good data, and I'm driven by the data. And that's what I think about all these variants. And hey, I could be wrong, but there's no data to prove it yet. I think that's right. And people like Ron and others also agree with me, but they don't talk to the press. And so the press gets the other story. So I went to the New York Times, Apurva, whatever her name is, Mandrilli. I wrote and I said, you got the narrative on the variants all wrong. And she said, well, you're the only person who feels that way. Really? Look at Ron Fouché's letter. I'm not the only person who feels that way. I can get others as well. But, it's cool. It's scary to be more transmissible, right? So the press loves it. I think they're doing a disservice, frankly. Okay, Faster Tortoise. You got me to rant. Well, I usually say for sure. Okay. Ask one of our listeners here, Steph SF, that San Francisco has hit herd immunity. That's what they say. We were talking about it earlier. HCH, thank you so much for your contribution and putting up with my rants and so forth. But I'm very passionate about this. I tell people who complain, they write to me and say, you don't know anything about lab origins. I said, look, I've been studying viruses for 40 years. How many people in the world do you think can put that after their name? I'm not saying I know everything, but I've done one thing my whole life. Give me some credit for that. Universal coronavirus, at least 5 years, I would say. It's going to happen, for sure. I agree. And remember, if you want me to answer it, put my name. Oh, here we go. You're a nurse. You can't believe other nurses refuse the vax. There are doctors in some states who say don't get the vaccine. It's crazy. It's totally crazy. They're supposed to be helping people. That's their job. I don't understand what they're thinking. Thank you, Justin, for your contribution. I really appreciate it. I will keep doing this as long as I can. And my local supermarket bleach is it's $6 for a gallon. And the scientific supplier, it's over $50. It's crazy. It's crazy. I just finished undergrad and will be doing COVID immunology research. Are there key papers you'd recommend reading? Yeah, stay away from the COVID ones. Go back and read some key papers. The ones we discuss on Twitter are generally good ones. If we haven't discussed them and it's in Nature, Science, and Cell, well, it could be that we didn't get to it. But also, I think a lot of the stuff out there just isn't worth repeating. But maybe on an upcoming Immune, we can address your question, key papers to look at. Can you talk about why some viruses are able to integrate and others don't? So, as you know, retroviruses can integrate because they encode an enzyme called integrase, right? And they also have a reverse transcriptase, which takes the RNA genome, makes a DNA copy of it, and then the integrase puts it into your DNA, and it stays there forever. Adeno-associated viruses do not have an integrase, but their genome can be integrated. It's not always integrated, only under certain conditions, by virtue of DNA repair mechanisms of the cell. The viral DNA looks like it needs repair, and it gets integrated as a consequence. And adenoviruses don't do this. Herpes viruses don't do this. Most other viruses do not. Now, there's one exception to all this. If you look at the genome sequences of living things on the earth. You can find bits of viral sequences in them. Bits, non-functional bits that have just accidentally gotten integrated over the years, and as far as we can tell, have no functional value. And we don't see SARS-CoV-2. It's probably too early yet, and I don't think if we ever did, it would be tens of thousands of years from now, because we're talking about integration on a evolutionary scale. These happened years ago, so they have no role in the function of the virus, in the reproduction of the virus, but they just happen accidentally. In the cases where integration is important, the best examples are the retroviruses. Even for the adeno-associated viruses, integration isn't essential for reproduction. The virus genome remains as an extra chromosomal plasmid for a long period. Pfizer is only 88% effective. Only 88% is damn good. And what is it? What do you mean by effective, right? Always look when they say 88% effective. Is it COVID? Is it all COVID? Asymptomatic, mild, moderate, severe death? You have to look at that. I'll bet it's 100% effective against severe disease and death if you can find those data. Because the T cells, and this is the other part of my rant, the epitopes that T cells recognize are not changed in the variants. And that's why the variants are not a concern to me in terms of disease. They are concerned in terms of using monoclonals as therapy, but not in terms of disease and death. The state of Tamil Nadu in India was denied ivermectin by Tamil's board because Gilead was kind enough to roll in with remdesivir. Okay, remdesivir, they got a good deal, $15 a dose, which is good. And it can have some effect if given early. It's an antiviral, right? If you're out of breath and you have to go to the ICU and get on oxygen right away, it's too late because your viral phase is over. Remdesivir is not going to work at that point. You have to give it right after the first positive test, and then it will have some effect. Thank you, Ditching the Grind, for your contributions. Live podcasts, live streams, more to come. That's the whole point of doing this studio. We're going to do more of this because I see that people are interested. We'll see post-COVID how much interest there is, right? But I'm going to keep doing it because I love it. Would love it if you made a page with recommended papers. Okay, we will do that for virology and immunology. And, more transmissible comments. I'm not going to... I've done my rant, okay? Futz with the Lutz. Yeah, I futzed with it. I can get a good image. I know how to make an image. Last time I just didn't do it in time. Are preprints still being published a lot? If so, will we be going back to full peer review process? Now, preprints are here to stay. They're all reviewed for the most part, right? We're just going to see preprints forever. It's always going to be here, and I think it's here to stay, unfortunately. But I think it's good because people have access to the research that they support with their tax dollars, right? The problem is the press gets a hold of them. They don't know what to make of it. They ask people their opinion. The people don't read them, and then you get the wrong opinion. That's what I think happens. To clarify, the simplest GOF study is just passaging virus in cell culture. So no need to go nuts, people. We must do this work or the world is more dangerous. Yeah, let's say I take poliovirus and it doesn't grow in some cell in the lab, and I pass it until it grows. That's gain of function. Pretty harmless. That gets done all the time. If you select a bacteria to be antibiotic resistant in the lab, it's gain of function. If you have an enzyme that makes beer and you want to make better beer and you change the enzyme, that's gain of function. So if you want to get rid of gain of function, no beer, folks, no wine, no cheese, none of that stuff. I'm being a little silly, but you get the point. Gain of function was given to the avian influenza virus transmission experiments by a number of contrary individuals about 8 years ago, and the press used it to describe evil experiments, and that's not the case. So don't say to me Vinny, should we do gain of function experiments? Because the answer is, if we don't, you're not going to have any research. Certain kinds of gain of function on coronas and avian influenza are regulated because they have the potential to change the pathogenesis and tropism of the virus. And that's fine. They're regulated. You have to ask, is the risk and benefit ratio correct? And if so, can we do this under the right containment? And we address all those, and we can do those experiments. And then I think that's completely appropriate to do these experiments under those conditions. That's a good question. In the ivermectin studies, do they control for patients with underlying parasitic infections? So I think, it depends where they're going to be done. Yeah, it's a good question. I'm not sure where they'll be done. They're going to be done under the active umbrella of clinical trials for COVID. Hopefully they'll address it, because I think that's an important issue. It may be that the underlying infections are actually what are being treated, right? Yeah. Thank you, Mr. Ozzy Cam. 2 papers I posted on TWIV outline the risk of MERS and SARS-CoV-2 recombining. MERS is mostly a camel virus with the ability of SARS-CoV-2 to infect people. Can you dismiss it as possible? I don't dismiss it. I'm not worried about the recombination. I think there's not a lot of COVID in the area where there's a lot of MERS. And as Amy said, the viruses are genetically distinct. Even though they're beta coronas, I think the likelihood that they can recombine is low. But in fact, Mr. Ozzycam, what do you want me to do? Say, yes, it can happen. We need to do something. We need to vaccinate the camels. And I am not going to vaccinate the camels. It's not my job. It's the job of the people in the Arabian Peninsula who own the camels. There's a vaccine for MERS coronavirus that works in camels. It just has to be deployed. I don't care that Delta is surging anywhere. If it's surging, SARS-CoV-2 ancestral strain would have surged as well, folks. Do you understand? It's just because there are non-immune people. Now everyone's out and about and they're getting infected. It's not a variant issue. It's just SARS-CoV-2. Stella, thank you for your super chat. What's your secret to being a PI and doing all the SciComm? Amy! Amy is the secret. I could not do it without Amy. She came 6 years ago and I said, I really want to communicate more, take care of business, and now it's really her lab. And I'm just an advisor. Virology nerds are the best. Please hit the like button, folks. Thank you, Stephen, for your support. Really appreciate it. To condense TWIV, adjust the playback speed. You can. People do 1.5 and 2x speed for TWIV. They say it works fine. Some people want it longer, right? And that's fine. So I hope to give you everything you want, right? Yeah, you would miss the chit-chat if it were edited. So you don't have to listen to the edited one. You could just listen. There'll be 2 versions, right? There'll be an edited and a non-edited. TWIV Express. That's cool. And yes, you should listen to my online virology course. I revise it every year in the spring. It's on YouTube. It's free. I feel that knowledge should be free to everyone. And I think it's full of information. It's updated all the time. You have a MedCram type of TWIV. God, MedCram gets so many views because it's short and it's what docs need, right? I understand that. And what I do is not what docs need. Oh, well. Earlier paper, no evidence of increased transmissibility. Instead, resumption of travel, lack of effective screening and containment may explain the variant success. Thank you very much. Exactly. That's what I've been saying all along. And then there's always people who say, I don't know what I'm talking about. You know, you can disagree with me, but just be civil. Oh, man, if I showed you some of the emails I get, so uncivilized. Why can't you disagree civilly? That's all I say. Hello. My sister has become anti-vax. Should I throw her off a building or lock her up? No, don't do either. Just try and calmly convince her by saying, you're vaccinated and your family is vaccinated, and it's really good. John Gruber suggests Twivbits. That's not a bad name, Twivbits. Twivbits. Thank you, John. On YouTube, you can easily timestamp the different sections. If I got a helper one day, I would do timestamps. Right now, maybe Les, do you want to do it? Send me an email. I know you wanted to volunteer once, right? Let me talk a little bit about volunteering, right? So I've had a lot of experience with volunteers and Microbe TV. And the experience is people say, I want to volunteer. And they start, and then after X amount of time, they stop. And I totally understand that they have other things to do. So I prefer to pay people to do things. And that way they stay. So if you want to help me, I'll pay you to do it. Because I don't want you to start and then leave. It doesn't help, right? And I could name so many things, like Kiki's comments earlier on Twiv. She didn't have time. She stopped. And you know what's the worst part? They never tell me. They just stop. All of them, not just Kiki, but all of them. They stop doing it. And I feel sad that they couldn't just say, hey, Prof, or hey, Vinny, I just can't do this. I got it. I understand. But not to tell me is sad. So sad. I have a Twitter page that only addresses SARS-CoV-2 and one other science. I promote Twiv and Dr. Griffin all the time. It would help for everyone here to promote us. I know there's some people here that don't like us. We get the fingers down. But you drop the name as much as you can. I would appreciate it. As I said, we don't make money. We just do this to educate people. I drop your name under YouTube and Twitter regarding promoting concept of humanity needing a cocktail approach. Mm-hmm. What was that? What was that one that was suggested? I have to write it down. See, I've already forgotten. John Gruber suggested it. Oh, I can go back and look at the chat. No problem. There's no excuse not to listen. Yeah, people have all kinds of excuses. I'm too busy. It's too complicated. I love listening to podcasts I don't understand because eventually I'll learn, right? It's really cool to do that. Twivbits. Twivbits. And now we have an explanation of POTS. Vincent, I live in the tropics, got flu all the time. It only stopped after I took the flu shot. Okay, thank you. Yes, there is flu in the tropics. Where can we find Vincent's virology course? Well, if you're watching this live stream, you are on my YouTube channel, youtube.com/profvrr. And right on the front page, there should be a virology course. If crap hits the fan, we have another pandemic. Well, we're going to have another pandemic. It'll probably be viral. The next pandemic is going to be an influenza pandemic within the next 10, 20 years. Twiv Light. Oh, so many good suggestions. Why don't adenoviruses integrate? I would say the real reason is there has been no selective evolutionary pressure for them to do so. They can cause persistent infections, and the mechanism of persistence is unknown. So in other words, what do I mean by mechanism? Why does the virus stay around? It doesn't seem to integrate. And it doesn't have the machinery to integrate. It's a double-stranded DNA that looks like the host DNA. Thank you, Sandy, for your contribution. Really appreciate it. Do you think a reason some are nervous about the mRNA vaccines is because they don't know the central dogma, don't know we have RNA in oral cells? RNA sounds foreign, maybe. But don't you think having tens of millions of people receiving these vaccines, and most of them are fine, 99.999% have not had any issues, wouldn't that work? I don't understand why they're not convinced. I'm not putting anything foreign in my body. Really. What do you put in your mouth every day? What goes in your nose? It's all kinds of foreign bits. They're excuses. I don't get it. So I'm not good at figuring it out. So we have nuggets. Orphate 9 changes mean what? Well, we don't know. No one's done the work because everyone's focused on the spike. Spike is sexy. Yeah, that's a t-shirt. Spike is sexy. Orphate could be an immune antagonist. Deleting it could make the virus less virulent and more able to spread among people because they're out and about, more asymptomatic infections, right? So we don't know. These need to be studied, and people don't study anything but spike pretty much. But now it's starting to happen, and we're going to see more and more papers. So what does virulence really mean? Virulence is the capacity to cause disease. You can measure it in different ways, but you could say how many people die is one extreme measure of virulence. For HIV, the CD4 count, how low does it go? It could be a measure of virulence. So there are many, many ways you can measure it. You just have to make sure if you're comparing them that you use the same measures. And how viral proteins influence virulence is a very interesting area, which we have worked on our whole careers in my lab, and many people do. Understanding how a viral protein or a change in a protein can affect virulence is fascinating, absolutely fascinating. Eurico. Hey, my buddy from Brazil. Good show, Vincent. Hi from Brazil. Full of COVID. Yeah. So Eurico was my host twice in Brazil when I went to do a pod. And here he is following the live stream. Good to see you. And let's get together one day again, Eurico. I'd love to visit. I always said the Brazilians were the nicest people. They're so friendly. Always smiling and nice to me wherever I went. It was great. Love to go back and do another pod there. This is an interesting question. How does the Italian wastewater sample of SARS-CoV-2 relate? I'm not going to say to the lab escape. It doesn't relate at all because there was no lab escape. But there's some idea now that the virus was actually circulating in people possibly in October 2019. And so it might have found its way elsewhere in the world. The problem with that study is that it's not clear if it was a contaminant or not. It was not the whole genome and so forth. So it needs to be followed up, and it hasn't been. But it's an interesting point. I didn't publish the email to the Chinese virologist. I just mentioned it tonight that I had written to her and assured her that many of us understand that it came from nature, and hopefully she felt good, and she was just lovely and gracious and so forth. Oh, this is very interesting. The intricacies of virology seem to evade even most doctors. It's misinterpreted by journalists and politicians. It's predictable. Public rarely gets a good service from these two. So I agree. Doctors have other things to do, and even keeping up, say, a surgeon with the latest surgical advances is hard because they have to make an effort. So to expect that they're going to keep up with other clinical sciences is unreal. It doesn't impact their practice in most cases. And the journalists don't have the training to understand the science, and they don't understand that if you talk to 10 people and 6 say one thing and 4 say another, you don't go with the 6. You go with both sides. You say someone says this and this. So the Times says they're more transmissible. I think that is a failing on their part. They shouldn't say. They should say some feel they are and some feel they aren't. There's a dichotomy in the field. But they have their sources that they believe in. Michael Osterholm, Peter Hotez, and so forth. Mark Lipsitch. And they go with them. And that is a failure of science journalism in my view. And this pandemic has made it very clear. And it's one of the other things that has made me very sad, that they can't cover it properly. And so we try to, and then we get criticized for being too long and too complicated and too dogmatic. But you know what? The 460 of you, 8 of you here, that's what I need. Yeah, Gribler's got the definition of virulence, which you can find in my lecture, a whole lecture on virulence and pathogenicity. You don't have to do lethality. You can do LD50. You can do fever. You can do weight loss. You can do a lot of things. Thank you for your contribution. I hope you all stay after the pandemic. After schools went back in March, a bump of rhino colds, recently a bump in paraflu. So parainfluenza is another virus. It is in the same family as measles viruses, paramyxoviruses. So parainfluenza are common respiratory pathogens that infect mainly young children, but also older people as well. And that's because we go back together and we're spreading our viruses. All right, let's go check through some of these super chats before we end up here. By the way, folks, I had a medical procedure today. I'm fine, but I had anesthesia. I was out, and I'm still here because I love you guys, and I wouldn't miss it. So I'm a little tired, and I'm going to end at 9:30. Thank you, Ro, for your support, and thank you, and good luck in India. I hope you stay safe, okay? Morris, thank you so much for your contribution. Post-polio news. What are the chances the FDA approves at least one vaccine for non-emergency use by the end of August? I think it's a good chance. I think Moderna is applying for full authorization, and I'm sure that Pfizer will follow. Thank you, Vanity Nutrition. You should all know Vanity Nutrition is one of our two moderators, besides Frank P. And Vanity took my virology course at Columbia this past semester while doing other work. So kudos to you. Joe, thank you so much for your contribution. Really appreciated. A lot of good questions here. Unfortunately, I will have to pass because we are out of time, but really good questions. Come back next week. We'll be here. I'm looking for the super chat so I can thank them. Thanks, Bill. Really appreciate it. I always tell you what I'm thinking. I follow the data. I don't follow the rumors. Paul, thank you so much. You and your colleagues have been more right than any other source of information and seem to be the only science in the public discourse who are willing to admit when you don't know and when you're wrong. Thank you. I will be the first to tell you when I don't know, and I will tell you when I'm wrong. By the way, 6 years ago, I was wrong about an issue in my blog, my virology blog, and I admitted it. And I couldn't believe the outburst of support from people. Oh my God, a scientist who admitted he's wrong. I have so much respect for you. That was quite a learning experience. And lots of other great comments. Sorry we're going to miss them, but they'll be here for replay. Thank you, Hoplite76. I try to be clear and tell you what I'm thinking for sure. I'm not sure I'm great, but I appreciate that you think so for sure. Mr. Ozzycam, thank you so much. If you ever do come to Australia, I'll buy your lunch. Yeah, I'm happy to have lunch with anyone, by the way. I've been to Australia once. I went to Melbourne, did 5 pods in Melbourne. I'd love to go back. I have friends there. And by the way, this week I talked to a journalist. I talked to many journalists all week, okay? And this guy said, do you have a student I could sit down with and go over some papers I don't understand? And I said, I'll sit down with you. I'll sit down with anyone. I'll give you my time. If you want to call me. I was walking past the lab the other day, and Amy said, there's a lady on the phone wants to talk to you. So I picked it up, and I talked to her about vaccines for half an hour because she happened to catch me in the hall. Otherwise, I don't answer my phone. I'm happy to talk to anyone. And any of you, of course, are welcome to join me to ask me to talk to you. Looking for the super chat. Thank you, Overjoyed. Really appreciate your support. 1 neuron from your brain. No, I'm not any smarter than anyone else. I just have passion for things. I learned a while ago that being passionate is really good and curious. Want to know when something works, right? I'm never bored because of that. I always have passion. I can always think of something that'll amuse me. Anyway, folks, that's it for tonight. I appreciate all of you coming. Great questions. Bring them back next week. If you're here before you go, do a thumbs up, please. That way other people get attracted to it and will come and check it out. Anyway, good night, everyone. Stay safe, get vaccinated, and see you next week.